And today we're going to be talking about comparisons of spike proteins in human SARS-CoV-2 versus animal SARS-CoV. In our project, we'll look at human and non-human genome sequences. In order to compare these sequences, we determine the origin of COVID-19 through the NCBI virus database. It is likely that the virus traveled from non-humans to humans, possibly through intermediaries such as a pangolin. Mutation rates are high in RNA viruses, so different regions will have different amino acid sequences. Currently, there are at least 7 species to cause coronavirus in humans, and I'm going to pass it off to Shia Fisher.
Thanks, Katya. So a surface glycoprotein is also known as a spike protein and is a sequence of amino acids that make up a precise section of RNA. In our project, we are comparing spike proteins in humans and animals. Figure 1 in the bottom left corner shows a cell with a few ACE2 protein receptors and the spike protein of CoV-2 at an attachment site on a cell's phospholipid layer. Viruses can only replicate from within a cell, so this attachment site is very important to understand. And I'd like to pass it off to Joe.
Thanks, Shia. The NCBI database can be used to study the novel coronavirus that causes COVID-19 and analyze what amino acid sequence binds the virus to the host cell. We analyzed 6 different species: viverids, tigers, trident bats, dogs, common bats, and cats. And the results are shown in figures 2 and 3. The cat and dog sequences were both found to be identical to the human sequence. However, the bat was the least similar to the human sequence, suggesting the existence of an intermediate host. As previously mentioned by Katya, the high mutation rate makes it difficult to research this virus. However, recent research has been gradually developing improved methods, which will hopefully be able to bring us to a functional vaccine in the near future. Now I'll pass it back to Shia to discuss the findings.
Yeah, so a few things as we close out here. Unrelated to genomic sequencing is a report about bats in Wuhan, China during the early transmission cycles. Now, it's been reported that bats were in hibernation at the time of early transmission, and that furthers the argument that specific path of transmission may not have come directly from bats. I'd like to give our thanks and acknowledge Elise Van Ginkel and Lyamichev for helping us with this project.